after two German chemists, Emil Fischer and Franz Hofmeister, independently stated in 1902 that proteins are essentially polypeptides, consisting of many amino acids. An attempt was made to classify proteins according to their chemical and physical properties, because the biological function of proteins have not yet been established. Proteins. Protein comes from the Greek word proteus, meaning primary or holding the first place. It is used to refer to the complex organic substances found in the cells of living beings. They are made out of nitrogen, oxygen, and hydrogen. They are also known as the building blocks of life. They are connected intimately with all chemical and physical activity which constitute the life of the cell. Proteins can be antibodies that aid in defense against disease and infection. They can be enzymes that catalyze metabolic reactions. They can also be hormonal enzymes that control bodily functions.
For today, I will discuss the 4 levels of protein structure. But before that, let me show these illustrated pictures of the level of protein structure, which are the primary, secondary, tertiary, and quaternary structure. Now that you saw these pictures, let's proceed to the discussion.
Protein structure defined as a three-dimensional arrangement of atoms in an amino acid chain molecule. Proteins or polymers, specifically polypeptides, form from a sequence of amino acids, the monomers of the polymer. Level of protein structure. The first one is the primary structure of protein, refers to the sequence of amino acid in a polypeptide chain. It is held together by a peptide bond that are made during the process of protein biosynthesis. The two ends of a polypeptide chain are referred to as a carboxyl terminus or C-terminus and the amino terminus or N-terminus, based on the nature of the three groups on each extremity. To make it more easy, just remember that primary structure is simply based on the sequence of the amino acid found in the protein, where this sequence determines the shape of the protein. If you replace just one amino acid to another, it will completely change the shape of the protein. So it means that the function and shape of a protein is just based on the sequence of the amino acid. Next is the secondary structure, described as the localized shape of protein. Secondary structure has two types, the alpha helix and the beta plated shape, where a helix is a right-handed helix with the peptide bonds located on the inside and the side chains extended outward. They are formed between the amino and carbonyl group of every fourth peptide bond. Next is the beta-pleated sheet, is a common motif of a regular secondary structure in protein. It consists of beta strands connected laterally by at least 2 or 3 backbone hydrogen bonds, forming a generally twist-pleated sheet. Tertiary structures refers to a three-dimensional structure of monomeric and multimeric protein molecules. The folding is driven by a non-specific hydrophobic interaction, the burial of hydrophobic residue from water, but the structure is stable only when the parts of a protein domain are locked into places by a specific tertiary interaction such as salt bridges, hydrogen bonds, and tight packaging of side chains and disulfide bonds. Or in the easiest way, it's just the tertiary structure of a protein describes its three-dimensional folding pattern with one individual subunit. And when you combine a multiple subunit, you create a quaternary structure. Quaternary structure refers to the number and arrangement of multiple protein
molecules in a multi-subunit complex. Example of this is hemoglobin that has 4 individual 2 subunits, 2 alpha subunits, and 2 beta subunits. So, the question is, how denaturation occur at level of protein structure? In quaternary structure, denaturation, protein subunits are dissociated or the spatial arrangement of protein subunit is disrupted. Tertiary structure denaturation involves the disruption of covalent interaction between amino acid side chains, non-covalent dipole-dipole interaction between polar amino acid side chain, and van der Waals or induced dipole interaction between non-polar amino acid side chains. In secondary structure, denaturation protein lose all regular repeating pattern such as alpha helix and beta plated shift and adopt a random curve configuration. Lastly is the primary structure such as the sequence of amino acid held together by a covalent peptide bond is not disrupted by denaturation.
Denaturation is a process in which proteins or nucleic acid lose the quaternary structure, tertiary structure, and secondary structure, which is present in their native state by application of some external stress or compound, such as strong acid or base, a concentrated inorganic an organic solvent, radiation, or heat. If proteins in a living cell are denatured, this results in disruption of cell activity and possibly cell death. Proteins denaturation is also a consequence of cell death. Denatured proteins can exhibit a wide range of characteristics from conformational change and loss solubility to aggregation due to the exposure of hydrophobic groups. Denatured proteins lose their 3D structure and therefore cannot function. Protein folding. Protein folding is a key whether a globular or membrane protein can do its job correctly. It must be folded into the right shape to function. However, hydrogen bonds, which can play a big part in folding, are rather weak and thus easily affected by heat, acidity, varying salt concentration, and other stressors which can be denatured the protein. This is one reason why homeostasis is psychologically necessary in many life forms. This concept unrelated to denatured alcoholic, which is alcohol that has been mixed with additives and to make it unsuitable for humans' consumption. Here are the illustrated picture of the effects of temperature. The effects of temperature on enzyme activity top. Increasing the temperature increase the rate of reaction. Middle, the fraction of folded and functional enzyme decreases above its denaturation temperature. Bottom, consequently, an enzyme of optimal rate of reaction is at an intermediate temperature. And here are the process of denaturation. First, functional protein showing a quaternary structure. Second, when heat is applied, it alters the intramolecular bonds of the protein. Third, unfolding of the polypeptides or amino acids.
Protein denaturation. Denaturated proteins can exhibit a wide range of characteristics, from loss of solubility to protein aggregation. Protein folding consists of a balance between a substantial amount of weak intramolecular interactions within a protein, like hydrophobic, electrostatic, and van der Waals interactions, and protein-solvent interactions. As a result, this process is heavily reliant on environmental state that the protein resides in. These environmental conditions are not limited to temperature, salinity, pressure, and the solvent that happens to be involved. Consequently, any exposure to extreme stresses, example are like heat or radiation, high inorganic salt concentrations, strong acids and bases, can disrupt a protein's interaction and inevitably lead to denaturation. When a protein is denaturated, secondary and tertiary structures are altered, but the peptide bonds of the primary structure between the amino acids are left intact. Since all structural levels of the protein determine its function, the protein can no longer perform its function once it has been denaturated. This is in contrast to intrinsically unstructured proteins, which are unfolded in their native state, but still functionally active and tend to fold upon binding to their biological target. There are some common examples. When food is cooked, some of its proteins become denatured. This is why boiled eggs become hard and cooked meat becomes firm. A classic example of denaturing in proteins comes from egg whites, which are typically largely egg albumins in water. Fresh from the eggs, egg whites are transparent and liquid. Cooking the thermally unstable whites turns them opaque, forming an interconnected solid mass. The same transformation can be effected with a denaturing chemical. Pouring egg whites into a beaker of acetone will also turn egg whites translucent and solid. The skin that forms on curdled milk is another common example of denatured protein. The cold appetizer known as ceviche is prepared by chemically cooking raw fish and shellfish in an acidic citrus marinade without heat. As we can see on the top, the protein albumin in the egg white undergoes denaturation and loss of solubility when the egg is cooked. As we can see on the bottom, paper clips provide a visual analogy to help with the conceptualization of the denaturation process.
Loss function. Most biological substrates lose their biological function when denatured. For example, enzymes lose their activity because the substrate can no longer bind to the active site, and because amino acid residues involved in stabilizing substrate, transition states are no longer positioned to be able to do so. It means that the denaturation process and the associated loss of activity can be measured using techniques such as dual polarization interferometry.
Loss of activity due to heavy metals and metalloids. By targeting proteins, heavy metals have been known to disrupt the function and activity carried out by proteins. It is important to note that the heavy metals fall into categories consisting of transition metals as well as a select amount of metalloid. These metals, when interacting with native folded protein, tend to play a role in obstructing their biological activity. This interference can be carried out in a different number of ways. These heavy metals can form a complex with the functional side chain groups present in a protein or form bonds to free thiols. Heavy metals also play a role in oxidizing amino acid side chains present in a protein. Along with this, when interacting with metalloproteins, heavy metals can dislocate and replace key metal ions. As a result, heavy metals can interfere with folded proteins, which can strongly deter protein stability and activity. Reversibility and irreversibility. In many cases, denaturation is reversible. The proteins can regain their native state when the denaturing influence is removed. This process can be called renaturation. This understanding has led to the notion that all the information needed for proteins to assume their native state was encoded in the primary structure of the protein, and hence in the DNA that code for protein, the so-called Anne-Vincenz thermodynamic hypothesis. Denaturation can also be irreversible. This irreversibility is typically a kinetic, not a thermodynamic irreversibility, as generally, when a protein is folded, it has a lower free energy. Through kinetic irreversibility, the fact that the protein is stuck in a local minimum can stop it from ever refolding after it has been irreversibly denatured. Protein denaturation due to pH. Denaturation can also be caused by changes in the pH. which can affect the chemistry of the amino acids and the residues. The ionizable groups in amino acids are able to become ionized when changes in pH occur. A pH change to more acidic or more basic conditions can induce unfolding. Acid-induced unfolding often occurs between pH 2 and 5. Base-induced unfolding usually requires pH 10 or higher.
A protein is a polymer consisting of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about individual structure of an amino acids. So an amino acid has a chiral carbon with a hydrogen attached to it. And it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid next to it. The left side where the nitrogen is located in the N-terminal of the amino acid. The right side that has the carboxyl group is the C-terminal of the amino acid. Now these two amino acids can react with each other in a condensation reaction where we're going to lose water and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called the dehydration reaction because you're losing water. And so, this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2. We have one hydrogen left over once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, and that is the peptide bond. We form a bond between the carbon and nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break.